Welcome everybody to today's seminar lecture, molecular structure biology. The topic today is signaling, and what signaling, what we can do in structure biology to contribute to the very difficult understanding how cells actually perform information processing, do react on certain environmental cues, do this or that. First of all, I think, signaling is a topic which is very difficult to teach because it is an almost endless story. Yes. So when you consider enzymology has a lot of different links, to genetics, to protein chemistry, and so on. Very complex. You can involve a lot of people from different fields, but signaling, that means the way how cells, whether they are bacteria or eukaryotic cells, process signals either from the environment or from the host itself or from intracellular regions, it's pretty difficult. And this show build I show you here is a very simplified one for some eukaryotic pathways, only some of them, about which I will give also some talk, not all of them, during this lecture. We may already recognize some good candidates. Stat 5. I think we are Stat 4, yes. We worked on this at the very beginning of the lecture during the exercises. You remember that was a DNA binding protein. With information processing, either from the environment, from the host, when you are, for example, a bacterium living in gut, or when you are a multicellular organism from other of your colleagues, from your other cells. So the cells are capable of integrating this rich amount of information and come to some certain kind of cellular response. And that kind of response, is calculated somehow by protein networks and also some genetic networks. So today we will focus at the beginning a little bit on the cytokines, and on the Janus kinase, but I will also give some rough view also on some of those other ones and what we can learn as structural biologists from signaling and what we can tell our structural biologists from our kind of experience. First of all, signaling is not only very complicated, but it's also dominated by protein interaction networks. So networks, as you can imagine, give some correlation, who is in interaction with what, who is in charge of what. So we know that in sociology, but also, in other sciences, these kind of networks describe a little bit, how these kind of different proteins, signaling proteins, in what kind of relationship they are with each other. For example, the epithelial growth factor EGF is binding to the epithelial growth factor receptor, and that is doing something with GRB2, an adaptor protein, SOS, RAS, and so on, and so on, and so on, and so on, till finally somewhen, we have a response happening on DNA level, for example, by promoting gene expression or promoting cell division. So protein interaction networks are very complicated, and the basic part of those ones, let's call them the central elements, are the protein-protein interactions themselves, PPIs abbreviated. So PPIs, protein-protein interactions, are also can be very complicated. You see this example here on the right-hand side with a kinase, this extracellularly regulated kinase called ERK, pretty important central kinase in eukaryotic signaling. And you see quite a few, maybe not all, of the known interaction partners which have been demonstrated by genetics, by physiology, by structural studies, that they all do interact with this protein kinase, and they somehow affect the activity and the context in which this protein kinase is acting. And that is a representative example. You can imagine now how that looks like when you have many components like those ones. There's also another problem. Protein-protein interactions in the crowded environment of a cell are numerous. So it's very difficult to have complete maps, what kind of protein-protein interactions are actually present in a given cell. So there are a lot of different experimental methods. We cannot come to those one in this context, maybe in biochemistry 4 in the methods module, like yeast to hybrid systems, phage display, protein arrays, where you just look, is it interacting with a given position on a protein array. Mass spectrometry is a wonderful method to get a lot of information about protein-protein interactions, fluorescence, resonance, electron transfer, and our structural methods like X-ray crystallography, cryo-electron microscopy, and so on. But there are also some other methods which we should not forget. For example, those ones which involve in silico docking, where you dock one protein to the other one, see whether it forms sensible complexes. Motif searches, which are done to recognize very often used motifs for protein-protein interaction. Genetical covariation, when you have a lot of genomes available. Nowadays, trying to use artificial intelligence, and so on, endless. And there are also some rich databases containing quite a lot of information about these protein-protein interactions. Please don't forget, protein-protein interactions and protein interaction networks are not only meaningful for signal transduction, but also for many other biological complex processes that we do know, even for example for translation by ribosomes. Okay, let's take a case example, to get a bit warmed up. Let's take the growth hormone. Yes, the growth hormone and its signaling pathway. So this looks a bit simpler now. Yes, the growth hormone, abbreviated GH, is binding on some receptor, and the receptor is undergoing phosphorylation, doing autophosphorylation first, starting to interact with a lot of components, change its conformation, and then starting to phosphorylate stud proteins like stud5, as we have seen it when we made a look on this kind of structure of a stud DNA complex and what the tyrosine phosphate is actually playing for. These kind of receptors, these growth hormone receptors, are one representative of the so-called cytokine receptors. Cytokine receptors are membrane-bound. They are homoheterodimers. Mostly the ligands are pretty small, or small peptides, or small peptide hormones like GH. They're different types. They are fused to something that is called the Janus kinases, or at least they interact with some Janus kinases. These are protein kinases which phosphorylated tyrosine residues by using ATP and transferring the terminal phosphate to a tyrosine on the selected sequence. Usually they have one that is active, and the other one is a pseudokinase, is a pseudokinase domain that is inactive and just used for regulation, for nothing more. In our stats, we already have seen a little bit, these are transcription factors that go to some gamma-activated sequence motifs in the genome of cytokine-inducible promoters. So that, was the way how they're activated, but let's go a little bit into the structural, more molecular details than just this kind of, let's say, simple representations. First of all, what is interesting, these kind of cytokine receptors are dimers, I said. The growth hormone receptor is a homodimer. So there are two identical subunits which form together the receptor, as somehow here indicated on the lower right. So the interesting point is, but they bind only one ligand. So there's a 2-to-1 stoichiometry for ligand binding. Two receptor polypeptides bind one polypeptide of the ligand, in this case the growth hormone. So that is interesting because it tells us something about biology and symmetry. Sometimes very often in biology we find symmetric complexes. Symmetry is pretty uniformly distributed. When you think, for example, about enzymes involved in glycolysis, they are very often dimers and tetramers, and these are homodimers, homotetramers, and all the subunits look similar or identical, or maybe apart when there is some allosteric regulation, as we have seen in biochemistry 1, for example, in hemoglobin or some enzymes. But what happens when symmetry is broken? And that's interesting. Sometimes symmetry is deliberately broken by special interactions. So that means that this polypeptide, this growth hormone ligand, is binding in a different way with this polypeptide than to that polypeptide. Also chemically, these two polypeptides are identical, and in the absence of a bound growth hormone, they formed a symmetric dimeric complex. So we will ask ourselves, what are the prerequisites for breaking symmetry by interaction? Another point why actually this is a very fancy example, it's a growth hormone. Maybe you see here, for what you can take it for. Maybe some of you have six-pack muscles, like this gentleman behind, Unfortunately, quite often people have to add some additives, during training and try to misuse this kind of peptide hormones to get that bit faster. I think recently there was a death, of one of these sportive people who took probably too much during his active time till he was dead, because there are, of course, some side effects when you use this kind of a hormone. But, you use also medicine. That's quite obvious. And the binding constant is known for the human growth hormone to its receptor. It's about zero 0.3 nanomolar, which is a high affinity interaction. Or when you want to translate it, something about 50 kilojoule per mole that you can get, from the interaction, from a one-to-one interaction of the growth hormone, one-to-one interaction with one of the receptors. But we have a two-to-one interaction, interestingly here. So be careful. So what is about these kind of receptor domains to be said? As you see here, these the receptor domains, they look somehow very similar. Indeed, the domains, there are two domains per receptor polypeptide, which are looking outside the membrane. They are from the same domain type. It's a so-called fibronectin type 3 domain. I guess we had them in the introduction about domains. It's a beta sandwich domain that is very common in extracellular proteins or also on surface exposed eukaryotic proteins, not bacterial proteins. In contrast, the growth hormone itself here, looks like a typical 4-helix bundle protein, which is also a very large family of proteins. You see, two different types, but what about the receptor domains? So when you look for eukaryotic receptors, also of signaling receptors, you find very often that the receptor domains, which are hanging outside the plasma membrane, are beta topologies. Fibronectin type 1, type 2, type 3, but very often type 3, furin-like cystine-rich domains, immunoglobulin-like domains, for example, receptor tyrosine kinase, also a lot of cell adhesion molecules actually use that kind of topology. You find this kind of topologies also in not only in mammal proteins, but also in fungal proteins. As we know from our own work, we found some fibronectin type 3 domains in a well-known fungal adhesin. So they are really, really numerous. So what about the asymmetry that we have said? It's interesting because you see when you look on this kind of heterotrimer, two polypeptides. Here's a growth hormone. You see that the majority of interactions are done with the blue polypeptide with the D1 domain and a little bit with the D2 domain. That's the second domain. There is also one interaction with the D1 domain of the other polypeptide. So what is possible to show is you can make mutants and analyze it. For example, you can mutate this glycine residue that is here in this interface to some residue that takes a lot of volume to break this interface. And you can nicely show that when you do that, when you add up a growth hormone that has this point mutation here to arginine, that you only get one-to-one complexes, not anymore these two-to-one complexes which are physiologically relevant. So this indicates you that this is somehow some additional interaction, but not important. When you break this interaction here at that site between the blue chain and the red chain, the epithelial growth factor, by making proper mutants here, you abolish binding completely. So the green-red interaction surface is not sufficient to form stable complexes. So, conclusion, we have a main interaction, and the main interaction is done just between a single receptor polypeptide and the other one. But the question is, why do we have the second interaction? That is a good question. So, what are people doing to understand this kind of protein-protein interaction between a receptor and its target? It's in this case a growth hormone on a molecular level. Clearly, you can make a look on structures, but structures will not tell you necessarily the thermodynamic relevance each position, each residue has for complex formation. So when you look on structures, you get mostly a static view where you can of course count hydrogen bonds, count electrostatic interactions, plus and minus arginine to glutamate or whatsoever, van der Waals interactions, that means packing, steric packing interactions, and so on. But it's very difficult, it's not trivial to say this residue is the most important residue in the interface, the other residue is not. So Clarkson and Wells did in the beginning of the research on the growth hormone a lot of mutational studies. And that was actually also part of the efforts of that time of the company Genentech, who was a leader in developing protein engineering and recombinant proteins for therapy, which used this kind of data to develop optimized polypeptide hormones as the first ones on the market, long before the big companies came and do this business themselves. So what one can do to understand interactions is to mutate every single residue to a different residue. And the most conventional change of mutation, of point mutation that you do is a mutation to an alanine. So alanine scanning is the standard method to find out which residues are important for some given function or some given interaction, which residues are not important. And that has been done. And you can imagine how much work this means in the biochemistry lab when you generate not only the wild type and study the interaction of the wild type protein, but you generate 40, 50 mutants as well and do the interactions at the same time as well. You see that done here for a lot of residues that have been mapped to the interaction between the growth hormone and the receptor, mostly here the D1 domain. What do you see? You see that the residues have different kind of buried surfaces. You know that by the structure of the complex. However, when looking what's the most important residues in terms of energetics, that means of loss of free binding enthalpy, which you can measure. You measure dissociation constant 1, you measure dissociation constant 2. You use the Gibbs equation to calculate delta G, for this binding or for that binding of the mutant. You calculate delta G to see the difference. You see that not necessarily residues which bear most of the side chains upon complex formation like this asparagine here to contribute necessarily to a large extent to the binding itself. This residue, this tryptophan residue, and this tryptophan residue, they are main contributors to the interaction sites, but there are also some other that is not so dominant here at that site, this prolin, that is also giving a lot of contribution, to the binding energy. So-called hotspots are those sites which are mediating most of the interactions also in energetic terms. And you see them marked here in red. Also, there's a lysine residue, lysine 172 for some reason, which I do not see here in this figure, which are contributing the most to the binding itself. It's interesting when you look on the distribution of these hotspots, they are found in the center of the protein-protein interface. So most of them are in the center. The other ones are mostly at the rim. That means when you have a protein-protein interface, look on the center of the protein-protein interface. Maybe you see some kind of dominant interactions like large bulky aromatic residues or some salt bridge which make up which make up this interface, whereas the other ones are only adapting to the interface itself. So here are some examples what kind of interactions you may find. We said on the receptor there were these two tryptophans, this tryptophan 104 and the other one, I think 169, that we have seen before. From the human growth hormone itself, it was this lysine residue that came up here, and the lysine residue is making a huge interaction with the tryptophan, with the indole side chain. And that is something that we often forget when we just look on our basic biochemistry textbook and see what's the role of the lysine residue. Everybody says clearly it's a basic residue. It has a protonated amino group in its side chain. It's positively charged usually, but one should not forget that it has a large aliphatic side chain as well, 4 methylene groups. And these 4 methylene groups here do steric packing with the indole side chain of this tryptophan. And in some cases, which are also known, you may even have some interaction between the amino group, especially the NH bond, with an aromatic side chain. That is a so-called p hydrogen interaction, where you have here the aromatic side chain. And here you have the bond between, for example, N, and here at the tip of my finger, the H, and that is interacting with the p electron cloud above the aromat. This is also an interaction that is often found, for example, in Erd Bremer's group here at the microbiology. This has been found to play a major role, for example, in the binding of certain kind of osmolytes by bacteria. But here we have a lysine to tryptophan side chain interaction, which is dominant. Not necessarily predictive when you just look on the structures itself, and you would wonder, is it contributing to the energy? You would say maybe it's some passive interaction. So the problem is, how do we recognize hotspots in protein-protein interactions, and especially when hydrophobics dominate over electrostatics? So how we can do so? Clearly, there are a lot of biophysical methods to study anyway these interactions, also of the mutants, And I will
Mr. Essen?
Yeah.
There's a question in chat.
Yes,
I just noticed.
just give the question now. Can you read it, please, for me?
Ja, ist immer auf Deutsch.
Ja.
Der Robert fragt, wie groß die angegebene Energiebarriere einzuschätzen ist, weil die Energieunterschiede von einem 6-Ring-Flip seines Wissens nach größer wäre und ob die dann bei Raumtemperatur trotzdem ablaufen, ungehindert.
Very good question. So you are right, So when you have buried aromatic side chains, the energy barrier, for a flip over of the let's say for phenylalanine, for example, which can do a flip over without causing a structural change in the environment after the flip, is very large. So these kind of flips occur on a time scale in the minutes range. I have a bit the problem, to give a precise or let's say a good estimate for a delta G value, but I assume these are values of more than 8 to 10 kilojoule per mole. Yes, so something that is proceeding very slowly. However, when considering now the interaction that we have found in this complex now, when this complex is formed, the tryptophan is on the surface of the D1 domain, also the other tryptophan. So what do we also see? That's the reason why this is a very good question. We have sometimes in protein-protein interaction, especially in protein-protein interaction sites, aromatic residues that are becoming part of the protein-protein interaction interface. So they are on the surface. They can maybe also do a flip upon the binding of the ligand, but because there's no steric hindrance, because they're exposed to bulk solvent, the barrier for them to flip over or to change their precise orientation is negligible. That means just very few kilojoule per mole. That means that happens upon complex formation. These kind of aromatic residues are very often found, for example, I'll give you another example, in the antigen binding sites of antibodies. Antibodies are well known to interact with, that they're capable, depending on the specificity, to interact with a lot of different antigens, just by varying a bit the structure. And very often we find actually tryptophans and tyrosines in these interfaces. So there is something that aromatic residues are not simply, um, um, let's say, hydrophobic residues like isoleucine or leucine, for example, because those residues would more drive hydrophobically driven aggregation. So I could tell you pretty long about aromatic residues and that they're a bit special in terms of the kind of distribution. So, but please remember, protein-protein interaction science, very interesting to look for those kind of residues. Very good question.
Thank you.
So, getting back to these techniques, I will not tell about these techniques so much, Maybe you should attend the Biochemistry 4, and I hope that most of the techniques will be somehow dealt with, and being talked about. I will just, in the context of this short talk for today, just go for some technique. I will go for surface plasmon resonance spectroscopy, But the other ones, it's not making this technique more interesting than the other ones. I'm personally a fan of ITC and MST, and also a fan of fluorescence titration, and dynamic and light scattering and thermofluor and biolayer interferometry, not so much of NMR. So it's just, for the sake of this kind of introduction, using the cytokine interaction as an example. So just in case you missed Biochemistry 4, that it was somehow being done over, surface plasmon resonance is a very interesting technique. It's a very sensitive technique. It's based on the fact that light is reflected by any kind of a metallic surface. Yes, for example, in this case, gold, which is good. Yes. So here you have already the setup of an SPR detector. You do reflection of the light. Yes, total reflection of the light here. Incoming, outcoming, and you detect what's going on here. The funny point is the angle of the incoming light is fixed by the machinery. Fixed. There's a prism mounted on it, usually here, to get this done here on the glass is a prism mounted to that, and the machinery, the incoming laser light is also attached to that. So in classical optics, you learn that incoming angle equal outcoming angle, and that is true when you have no quantum effects happening at all. However, in the case of this experimental setup, we have a very thin gold layer that is here printed on the, or evaporated on the glass, very thin gold layer, to which covalently attached is a matrix. And this matrix, mostly it is comprising some dextranes or related polyglucans. That one can be used then to couple, for example, some receptor protein, some ligand protein, or some ligand compound covalently to the chip. So what's happening during the experiment itself, during the classical SPR experiment, is you have a microfluidic setup where you have up your probe in liquid, water, buffered water, of course. You have here your detector where you modify the surface. And when something is binding up here, it is changing the mass, that is attached to this gold surface. And because, I hope that, is it on the next slide? Yes, I gave it on the next slide, the physical background. Very good. Give the background here, because something is changing, something changes with the angle of the outgoing light. So you see here, incoming, outcoming, yes, when you have a certain kind of boundary condition and you have an interface between here two different medium, let's say here glass, here mostly the gold, yes, you have here some interaction that is causing an evanescent field that is emanating here from this interface out. And any kind of disturbance here, where I have my cursor in, will affect the evanescent field. And the effect it makes on the evanescent field, or this plasmon, will affect actually the precise diffraction angle or reflection angle of the outgoing light. So here somehow indicated more, yes, incoming light causing here along the length path of the wavelengths, yes, lambda, this kind of dipole formations here in this kind of interface, and causing also to emerge here into the outgoing, into the lower part of our interface. So when you put up now when you change now the mass here by binding something to the receptor or to what is immobilized there whatsoever, you have a change to the evanescent field at that position, and that one will change in the outgoing angle. That is cool. So there would be two ways to measure that. You measure the outgoing angle, how it has changed. You can do that with some experimental setups. I think, for example, Nova Thumb did sometime with some own-built SPR machines, some measurements like that. Or, as you can also do, you're not measuring the angle, you're measuring at a fixed reflection point, the intensity of the incoming light, and you will see that then the amount of light will change, depending on how much here actually is loaded and how large the effect on the evanescent field is. And there are a lot of different chemistries available, so that you can even also couple lipid bilayers or something like that to this kind of chips and do this kind of experiment. So here again, yes, so what do you measure, when you do the experiment? Here again, the setup, here you see the change, yes, of intensity depending from the angle before and after, but most machines use a fixed angle setup. So you start with given ligand, for example. So here are your receptors waiting on the surface of the chip. Then it's the microfluidics. The microwave changes its position. Now you let the liquid flow. It's the same liquid, but now with the ligand. You see now over time, here in minutes, some change in the resonance unit. It's an arbitrary unit that is given by the instrument and how it's calculating the change of intensity here. A rise is made with a saturation point. So the saturation is indicating that your surface is now saturated. From the rise time, you can measure the binding constant, the K on of the formation of the receptor ligand complex. Then when you have the saturation point achieved, the machine is switching again the valve and is going back to the base liquid you had before. And now in the absence of the ligand, here shown in blue, you will see that now dissociation can happen now, because nothing is in the flow through now anymore of the ligand's presence anymore. So dissociation can happen, and you get a lowering of the signal. And that is giving you the dissociation rate of the complex, the K off constant. And the ratio K off and K on, that is actually pretty well known as a dissociation constant for a simple one-to-one complex. So from both rates, you can measure also dissociation constants. So at the end, when you have done this measurement, you regenerate everything by letting some buffer running through with some harsh condition that is disrupting all the kind of complexes, and then you equilibrate and do the next experiment. Pretty nice technique. You can do that at the Max Planck Institute. Machine is pretty expensive. Laser has to be exchanged every few years. Costs also 100,000 quid about. I think already this kind of change, it's giving you some quantitative information. We have now some techniques which are similar but more cost efficient than surface plasmon resonance techniques, which come up to some similar kind of data. So what about our epithelial growth factor receptor? We were talking about that this was still, we did still not address what's going on, you know, with this dimer, you know, this homo and heterodimer. So people did a lot of kinetic measurements for the binding using this kind of technology presented before. So what they did, they immobilized the receptor, the monomeric receptor, they immobilized it on the chip. Then they gave the human growth hormone to it. Then you see here this very nice sharp rise. Yes, you know that it's making the one-to-one complex. And then they added up, you know, now to this complex, now the other receptor part that is present here that can now bind in addition to the to this binary complex, and you can make a concentration dependent curve, and from this you can then also determine binding constant for the binding of the second chain, which was not possible before by using this kind of experiment, and by making the same strategy of making mutation to see which residues in the are now important for this secondary interaction with this other side. And what they found was, you know, they made some classifications. They found some binding hotspots, of course, in the growth hormone binding site itself here, yes, but they also found some hotspots for the interaction of these two D2 domains that are present. So when you mutate something here, it will disrupt the formation of this ternary complex, indicating that for the physiologically relevant complex, you have also to have this interaction between the two D2 domains which contribute to the binding at the other side. It's a very allosteric effect that you do have. And one can assume that makes sense, because when it would be only located here, the binding effect, we may have only a minor effect to everything that is attached to the membrane and going through the membrane to the cytosol, where at the end our receptor tyrosine kinase, or Janus kinase at least, is waiting to do something. So the signal has to be transmitted through the membrane. So this is indicating that this is the interaction site that is transmitting also the signal through the membrane. Cool. So there are a lot of other information known about other cytokine receptors. I like this picture very much, you know, here, because some of these studies are, of course, relevant for human sports, you know, like the kind of work done on erythropoietin, you know, and the EPA doping here that has been done, you know. Also, all the hotspots are actually mapped on the receptors, which are the residues. So you could think about synthetic EPOs, you know, by mutating around to the non-essential parts and doing this or that, you know, and to make a better Tour de France happening. But the problem is always, you know, after a few years they find it out anyway, because analytics is pretty quick to get it in after some time also to be fetched, and the urine samples of these poor people, yes, they are stored, yes, you know, for eternity in the fridge. So you will find them all, the doping guys, yes, who made bullshit with erythropoietin, you know, here interleukin-4 alpha receptor, something different. I told you something, that this is a fancy way, you know, alanine scanning, and to make these mutants to find out where actually contributions to the energetics are made for the binding. But unfortunately, the situation is not so trivial. So when you come up with the idea, you would do the same as they had done it for the growth hormone at the beginning of the 90s and end of the 80s. You know, we know much more about these systems. And we know that alanine scanning is very often leading to misleading results. Why? Naja, look on the energetics. So this is the wild type situation. This is delta G for the wild type. Fine. When you make a mutant, an alanine mutant, and you change something here in this mutant there, yeah, okay. There are two ways what can happen, yes, you know. First thing is, nothing happens here on the unbound complex, you know, the energy of the unbound complex is the same one as that one of the unbound complex of the wild type. no major change, maybe no major change in the kind of hydration energy and so on. There here this white square protein made after the mutation, nothing changed so much about this one here. In terms of the action with the bulk solvent, so you see a real change, yes, in the binding energy when you measure its delta G of a complex formation, you compare it with the wild type. Fine. But most often this is not the case. When you, for example, make an arginine to an alanine, you also change a lot about the hydration and the interaction with the bulk solvent. So it could also be this extreme case. You make all the changes, here in the ground state, yes, in the unbound state, yes, just by making the mutation, not because it makes some large effect because of the interaction formation. The reason is here first the interaction with the solvent here has changed, but not necessarily so much here within the complex. Remember, what does it mean I bind, these two partners do bind some to each other? It means when they're here in bulk solvent, the black one and the white one, they get rid of some part of the bulk solvent to which they make some interactions in the complex. And when I change something of the energetics with the bulk solvent, I change, of course, also the binding enthalpy, these complexes have. And in the general case, you change in both ways, yes, so you cannot make any kind of clever conclusions, yes, what this really means. So that is a problem, at least for some kind of receptor ligand pairs where for especially flexibility plays a role, strong electrostatic forces play a role that you also have then with water. So then this kind of delta G values may not mean necessarily tell you a lot at the end. There are also some other reasons with alanine scanning, yes, when you are interested to make them for quantitative analysis. There are many effects the change of a residue to an alanine can actually do. So that is a wild type here. So that is cool. You maybe switch off here this charge by mutation, this positive charge, and make it neutral, and you find some change there. Okay, fine, yes, cool, yes. But sometimes it can also something like that happen. The structure of the complex looks completely different, or looks already different by switching off some of the electrostatic interactions here. Or other example, you make here a large residue, like a tyrosine residue to an alanine in interface. What does it mean? Naja, when the complex is formed, you have a lot of water in it. So the structure is also not maintained anymore. It's a change of structure. So dealing with energetics can be difficult. Okay, clearly destroying all kind of interactions, by doing some change here is anyway a bit mysterious. Yes, you can get aggregation sometimes by making mutants. That's very cool. Which you can also get sometimes a loss of the homogeneity of your ligand when you make a mutation on the ligand. Maybe it makes a lot of conformations, or sometimes it gets even degraded. By being especially sensitive to proteolysis, which also happens in some cases. Actually, you can observe this in the lab. So there are some caveats to keep in mind for alanine scanning. This topic we already talked about when we had made a look on the stud DNA complex. Do you remember? After our cytokine receptor somehow got a phosphate group on the tyrosine of the studs. They changed their quaternary structure. They formed this nice tail-to-tail complex where actually the SH2 domain is interacting with the phosphotyrosine residue crosswise, getting up the framework of interaction, for the stud domains and making them suitably positioned to bind to DNA. So that was the basics that we have already learned. So just to remember what happens when you are not phosphorylating the tyrosine. So when you're not phosphorylating the tyrosine, the conformation is a completely different one. A different quaternary structure is assumed is taken by the stud proteins, where you then see that these kind of beta sandwich domains, they pack between these alpha helical domains. So these beta sandwich domains are here, those ones in red, and the alpha helical ones are the yellow ones, and now they pack to each other you know, this one with that one, this one with that one, and then the other domains here at the very tips, with a very large protein-to-protein interface. And this is also to remind you what is a large protein-protein interface. So we give them in square angstrom, and in general you can assume that meaningful protein-protein interfaces would be in the range of about 500 to 700 square angstrom and more. So 1600 is certainly meaningful. However, when you have an interface, let's say of 800 square angstrom or 700 square angstrom, for example, by solving some crystal structures, it is not necessarily meaning that this kind of interface is physiologically relevant, because sometimes you have also crystal interfaces which can be very large, a few hundred square angstrom. So PISA, protein interaction server, which we wanted also to use somewhere in the exercise, but we didn't do so yet, is a method to analyze protein interaction sites and the interface, and to see whether they are relevant, physiologically relevant, or they are dictated by crystal packing and some artifact interaction. 1600, however, is very large, you know, so there's no question at all that this interaction site is physiologically relevant. And before, we had also interaction sites with our hormone and our receptor for 1300 for a 1-to-1 complex, and about also 1800, you know, for the 2-to-1 complex. So, what about this kinase issue? Let's go from the extracellular part where we have the binding of something happening, yes, you know, just as we had discussed before, to the intracellular region now, where we have the effectors happening. In this case, in our EGF receptor, some tyrosine kinase domain that is being present there. There's a huge class on receptor tyrosine kinases known. This is a class of tyrosine kinase which is membrane bound, which has a receptor on the extracellular site. One of the famous ones that we all do know is the insulin receptor, because it's mediating the response of insulin to our cells, a very important one. And there's some diabetes known where there are some defects present in the receptor itself. The other receptors are mostly important for developmental regulation, like the neural growth factor or the fibroblast growth factor receptor. More than 60 classes, we cannot talk about them all. It's a long lecture. Let's just talk about how are they activated and how are they inactivated. Because these kind of tyrosine kinase are the effector domains. You have a signal coming from the outside. Okay, something happens in the receptor. You get a transmission of the conformation transmission through the membrane to the kind of domains that are in the cytosol, and then they change from an inactive state to an active state, or vice versa, depending on the type. So how this is achieved? So we see here we take first some very simple example, some soluble kinase, also tyrosine kinase. Let's take a very simple example before going back to our receptor tyrosine kinases. Let's take cyclin-dependent kinases. Cyclin-dependent kinases are most important all for every kind of eukaryotic life, because they control cell division. So the cyclin-dependent kinase phosphorylates proteins as other ones, but it needs the interaction with other protein components, the so-called cyclin here, yes. So the cyclin-dependent kinase complex is active and can start to phosphorylate other proteins. And the regulation occurs by the formation of the cyclin. And these are here dependent on the very different cell cycles. We have different cyclin-dependent kinases, 1, 4, 6, 2, and so on. You have different cyclins, A, B, C, D, whatsoever. I will not go into the details. They play a role in certain phases, you know, of your cell cycle. Some of them in the M phase, some of them in the G1 phase, some of them in the synthesis phase, and so on, you know. Doesn't matter. Look into biology textbook. You know how this is done in detail and why it's so super important for our mitosis. on the base of the protein, or on the level of the protein, we have an inbuilt inhibitor in our cyclin-dependent kinase. So the inhibitor is built in. It's here this blue region that is actually this blue region that is here interfering with a crucial residue here, this lysin, which is distorting, not being present here in the active site. So it has somehow to interact here with the lysin to get it into proper position, and to allow that actually the ATP can transfer its terminal phosphate to anything else, and also to open up the site because everything here is closed. So when the cyclin here shown in yellow binds to the cyclin-dependent kinase, it takes off this kind of helical element. It repositions everything a bit. You see the most structured chain occurring here in the blue region. That restores the active site. The ATP is still bound, but now also the site is open, that the substrate can get in with its tyrosine residue and accept the terminal phosphate of the protein itself. And you can already predict clearly these interaction sites here between the cyclin and the cyclin-dependent kinase. You can study them by mutation and do this and that to get that out. So it's a very simple mechanism. And when you degrade cyclin, which is the way how actually the cyclin disappears, it's inactivated by degradation. The cyclin-dependent kinase gets inactive again. So opening up of the active site, and this is a general principle that we find in almost all of these kind of kinases. So here are some other non-receptor tyrosine kinases, not only the CDC ones, the CDCs we had before, the CDC kinase, the cyclin-dependent kinases, SARC family, YAC family. Okay, we already had something. There are also soluble candidates, yes, which are not membrane attached, and this BTK tech ones. So I see, that is somehow to remind us, everything is so complicated. So when you see here, a common motive is a kinase. Cool. But what the hell are the other domains for? Especially these eukaryotic proteins, they have a lot of brain by having a lot of domains which form specific interactions. So the YAC kinase has this pseudokinase domain. They have this SH2 domain that binds to the tyrosine phosphate again. Yes, they have this kind of a firm domain. It's also an adapter domain. These other ones have pH and SH3 and SH2 domains in addition. All of them have multitudes of cytosolic additional domains besides the catalytic domain itself. And these are important for regulating the kinase activity. So intracellular binding domains, there are also certain kind of classes. There are also different kind of protein types. Some of them have been discovered, in a special protein kinase, in the SARC protein kinase, SRC called, yes, SRC. That was discovered, I think, in the 70s. There are two domains, SA2 and SA3. The pH domain, plextrin homology. Plextrin is a strange protein from red blood cells, but that has been discovered there, this domain. It's very, very often found. Firm domains, we had C2 and C1 domains, WD40, 7-bladed propeller domains, we have seen already in the introduction, and the F-hand, we have also seen also in the introduction. And these are just the most important intracellular binding domains for something. For calcium, for phosphotyrosines, for membrane association, and so on. So these are the signals that are processed intracellularly. Let's make a look. You see, we make some little excursion out to these domains. So let's make a look on the pH domain, on the plextrin homology domain. Very often found. Its function is mostly, but not always, it's mostly the recognition of lipids, especially of phosphoinositide lipids. Phosphoinositol bisphosphate, PIP2, or phosphoinositol trisphosphate, PIP3. These are themselves very important second messengers in the eukaryotic signal transduction machinery. However, there are also pH domains known that are not binding to PIP2 or PIP3. They bind to some other proteins, like the B-gamma subunits of G proteins. Mostly they play a role in the localization of any given signaling protein to the membrane or to complex formation. So what's funny? We can make a look in the protein data protein domain family, protein domain database. It's 1849 is the entry. And we see how many actually are found in eukaryotes. So this study is about 3 years old. There have been 33,000 proteins being matched. That means by sequencing, they have been found somewhere. So they are very widely spread, these pH domains. So because there's so many signaling proteins in every given organism and in all organisms of the eukaryotic clade. Second point is they are like Lego bricks, these kind of signaling domains. You find them in numerous domain organizations. You see here of these 2,137 different domain organizations, only a few of them with the order of domains. So they are often C-terminally, but sometimes N-terminally, sometimes in the mid, sometimes being part of larger proteins, sometimes of small proteins, even sometimes of pH domain only proteins. So an endless number of domains. So a common characteristic of all these kind of domains that we do find in signaling events is a kind of Lego-like assembly of the signaling protein. So they get the kind of complex characteristics by passing them together. Let's look whether this is generally true. The SH2 domain, at the beginning discovered in this SARC kinase, is a domain that recognizes the phosphotyrosine. We have seen this already in the STAT3 DNA receptor. The phosphate group itself is recognized by arginine residues. So electrostatic interaction is making the most of the interactions here. There's also lysine residues. So 2 to 3 basic residues at least, are surrounding this phosphate group, of the phosphotyrosine. And also, of course, here with the aromatics side chain interaction of this lysine residue. So it's not only electrostatics, but also some recognition of the aromat here. So this is generally done by SH2 domains, which recognize a motif like tyrosine phosphate, like this one here, for example, and then it has a glutamate in it. It's a very simple fold. It has a very high affinity in the low micromolar to the nanomolar range. And also here, 11,000 different proteins that match 640 domain organizations, numerous organizations, and most of them, almost all of them, are in signaling proteins. Let's finish with the SH3 domain. The SH3 domain recognizes polyprolines, especially 3 or 4 prolines in a row, or interrupted by a single residue. So these prolines, which are these here, which I show by the cursor, actually form locally a polyproline helix. You'll remember maybe from collagen that we have other secondary structures when the only proline is present in the polypeptide. It's not forming a conventional alpha helix, it cannot do so. It forms secondary structures of its own. And this one, this kind of polyproline helix that is recognized by the SH3 domains. The affinity is not very high, higher micromolar range, but sometimes you have one domain, one SH3 domain after another one, or just in combination with some other binding domains. So they can interact especially with non-structured regions of proteins, where actually simply several prolines are encoded in these non-structured regions to instruct then this protein to recruit other proteins that may have, for example, an SH3 domain. How is it here? 27,000 entries, 1,700 different domain organizations. It's the same as we have said it before. So these can be made in a numerous context with pH domains, with other signaling domains. Here, for example, in this example, where the SH3 and SH2 domain have been taken from, you see here's the kinase domain itself. Here's the linker. Here's the SH2 domain that recognizes the phosphorylated tail, the C-terminal end, of the SARC tyrosine kinase. So this is autophosphorylated by itself, or maybe also by some other kinase. And when it's autophosphorylated, the SH2 domain binds here to the phosphotyrosine in the same polypeptide. and stabilizes by this the inactive conformation of the kinase here. So that is one off. So when you dephosphorylate this site here by a phosphatase, a tyrosine phosphatase, you activate this kind of tyrosine kinase, which is also the regulation mechanism, or one of the regulation mechanisms for this protein. In the SH3 domain, you can already predict what it's doing. Here are some prolines in the linker region itself, which stabilize the interaction, the defined structure of this protein. Let's go back to our receptor tyrosine kinases, where we started at the beginning with the EGF receptor. Yes, let's make a look again to those ones. We didn't finish entirely because there are also some other points, which I have sometimes forgotten. So the epithelial growth factor has more than one signaling pathway. Another problem of our signal transduction friends, yes. So we talked about the STAT pathway, pretty nice, yes. So here something binds, it starts to phosphorylate itself, yes, it phosphorylates the STAT, yes, fine, and the STAT is making something in the nucleus. But the other pathway, and there are two other pathways, not just going about this pathway, goes via adapter proteins like GRIP2 or SOS on the MAP kinase pathway that is also going to this to the nucleus. So here about our epithelial growth factor receptor, we have seen it before already. This is epithelial growth factor. Now we talked about the growth hormone receptor. Sorry, I mix it a bit up. But the EGF, the epithelial growth factor receptor, you see it's also going via the STAT pathway. It can go like the growth hormone receptor by it. So one point is the extracellular part is different. It does not look like what we have seen before. It has 4 domains, pretty complicated. It forms a dimer. Yes, when it's bound to EGF, to the epithelial growth factor. You see this here, small polypeptide hormone. And you see this is again different. So we have here 1-to-1 complex, but now as a dimer made. Yes, and what you also see is that when you have the monomer, that is a non-functional form, but upon the binding of the EGF, it forms a stable dimer. And this is a clear allosteric effect. So the binding bit of this EGF between the domains 1 and 3 cause the domains 2 being properly positioned that they can interact with each other, and that they form then this kind of dimerization site. And by this, the whole receptor dimerizes here at domain 4 that is going to the membrane that's not shown here, and causing dimerization intracellularly. A different example how you can use binding of a ligand to its receptor to cause dimerization. In this case, clearly allosteric, not by sticking together to receptor ends. So the EGF receptor works in some way like the cyclin-dependent kinase receptor. So we see here also some activation loop, here it's called activation loop, that has to be properly positioned, the insulin receptor, which belongs to the same class of proteins, you see also known as structure of the inactivated form that is causing the active site not being properly formed. Here you see some inhibition coming in by some side chains and the interaction therein. So let's focus a little bit more what's happening here. So we have talked about SH2, SH3 domains. What is going downstream of this kind of receptor tyrosine kinase when they go via these adapter proteins? Yes, GRIP2, here in this case it's ERK, whatsoever it is, These proteins, this signaling pathway goes to small g proteins. So small g proteins bind GDP or GTP, and they can hydrolyze GDP to GDP, or they can exchange it. And they are in most cases active in the GDP bound form and can do something in the cell. So how is it looking like? So the first small g protein that was characterized is P21-RAS. RAS is a monomer GDPase. It is very important in oncology because it's a famous proto-oncogene where pretty often one can find mutations in later stages during the tumor generating pathway That means that sometimes mutations show up in P21 RAS to stabilize tumor cell lines in terms of their permanent growth. And RAS is a classical molecular switch because it has two switch positions. One is the GDP bound form and the other one is the GTP bound form. And it looks very similar like EFTU from translation or elongation factor G from translation or many other kind of G proteins. So the reason for that purple for the switching factor is here GDP, the nucleobase is similarly bound, also the sugar, but all the kind of conformation change is triggered by the terminal gamma phosphate here, the terminal phosphate in here. That interacts with these two switch loops, especially the switch loop 1, but switch loop 2 is also affecting its conformation by the presence or the absence of this terminal phosphate in the guanosine nucleotide. So the P loop is a third loop that is present here. This loop is actually pretty generic. That one you find in most ATPase and GTPase of all kinds. It has just two glycines separated and a lysine residue being positioned here over the beta gamma phosphates, and the serine residue, making some hydrogen bonding interaction with the beta position. This one is a very conserved loop. You find it all as a P loop. But for the signaling function of all this kind of GTPase, the switch loops are the important ones because they change their conformation upon GTP hydrolysis or transfer. Here you see that again, schematically, yes, RAS exists in two forms, inactive and active. And there are two ways to move, either by spontaneous hydrolysis of the GDP in the bound side of the RAS, but that goes very, very, very slowly. That takes hours to days. However, when you add up a GTPase activating protein that forms a complex with this RAS GTP, that catalyzes actually the hydrolysis of GTP to GDP and the release of the phosphate. So the GAP is an activator And the GAF is a guanine nucleotide exchange factor. This is actually like a chaperone for allowing a fast exchange of the GDP against GTP, which is in surplus in the cytosol to form the active state. This one is also highly regulating. Some proteins work as GAPs, others as GAFs. We will see some GAFs later. So switching is controlled by these two classes. So this is a damp pull down stuff. And RAS promotes receptor tyrosine kinase dependent cell proliferation and differentiation. And RAS, made to mediate of cancer. I mentioned that. 30% of human cancers. So let's try that we are happy and do not get this hyperactive forms of RAS working in us, So how is RAS working? Actually, because it's so important, it's intensely studied by a lot of different techniques, yes. So first of all, how is the hydrolysis done? Hydrolysis means we have a water molecule, an incoming water molecule, to the terminal phosphate of the GTP, and that forms a classical inline attack, as we know it from all the kind of other phosphoryl transferring reactions we have seen in biochemistry. It's inline attack with a bipyramidal trigger pentagonal bipyramidal geometry. You see that here with the spontaneous hydrolysis, and you see what happens when we have a GTPase activating protein present. This water here is exchanged by a guanidinium group of an arginine. So actually the catalytic group here is the arginine that is presented by the GAP into the active site of our P21 RAS and on the bound GTP and neutralizing the negative charge that is developing here on the phosphate or on the pyrophosphate group. And that is accelerating attack of the water that is also being a bit pre-adjusted by the switch 2 region, which is also here then affected by the interaction with this gap in its conformation. So that's pretty cool. Very simple switch. So how did one come to structures like that, How could one do actually kind of getting some idea how actually these structural transitions do happen. So for chemists, interesting is the point that artificial substrates, which can be manipulated by simple physical tools like light, are very useful. For example, GTP can be photo caged, by fusing it to this this nitrophenol-like compound here. You see this here on the left side. That is, don't ask me now for the systematic name. I would have to think too long about this. But you see that here the terminal phosphate group of the GTP, the Y would be the GDP, would stand here for GDP in this example, is esterified, to this kind of compound with this nitro group. And when you put near UV light on it with a rate of larger than 10 to 5 per second, at least when you go under saturating conditions that every single molecule is activated, so you get a very high turnover. You form then this activated form, and you get the photo decomposition, of this photo caged compound to free GTP and the remaining compound, here in this case a nitrosophenyl compound that is left over. So the idea is then very simple. When you would like to study processes like that, which go on, for example, in milliseconds or seconds, and you would like to do them structurally, you can use photochemistry to synchronize for example, in a protein crystal, but also in other experiments set up NMR, or maybe nowadays we can also try that in cryo-EM by using some photocage compounds to synchronize the reaction and then to follow on structural snapshots going then one after the another one, or trapping them. I have to admit, in this example, it didn't work. Yes, when they tried this to do here, this is some work of Alex Scheidegg when he was PhD student in Dortmund. He got nice complexes of the crystals, of these cryotrap compounds, but the cryotrapping itself was not going properly in the crystals without disrupting the crystals themselves. But in some other systems, it has been done and proven. So to see the interaction between the GTPase activating protein and RUS, I would like to remind you that the P21, actually the name says it has just a molecular mass of probably 21 kilodalton. It's a very, very small protein. That actually all these other proteins, they can be very large. It can have molecular mass of 80, 90 kilodalton. You see here one of these GTPase activating proteins is much larger than RUS itself. It's forming this complex. Its only purpose here for the reaction is to put in this arginine finger to the gamma phosphate to trigger the hydrolysis, of this kind of compound. And actually, you can ask how the structure was made, because we just learned when you like to co-crystallize something with, for example, GTP. After a few hours, everything is anyway converted to GDP. That will not work. So the miracle here was that actually chemistry is a quite good thing, and that we know analogs which behaves chemically pretty similar like the gamma phosphate by filling out the proper positions and forming a complex with GDP, but they are not phosphate. So example here, in this case, you see here this a few white atoms here, which are not further indicated here also, these ones. The central atom is a beryllium, and the distal residues, the three distal ones, are fluoride atoms. It's beryllium fluoride, BeF3-. This is a very stable complex, and this stable complex has a tendency, when you soak or when you add this up to a nucleotide like GDP, that it forms a coordination interaction with the oxygen group of this beta phosphate. So this is here a coordination interaction. And by this, it looks pretty similar like this transition state. I remember this pentagonal bipyramidal state there would be, of course, when this was a phosphoatom, and this was an oxygen, this was an oxygen, this was an oxygen, the other one would be here. The incoming water would be here where my error is, of course, behind the blue loops there, yes. But this is missing in the BeF3 minus complex. So these are structural analogs, that can be used to study structures of phosphoryl transferring proteins, which are otherwise difficult to get. Clearly, there are also other ways to do so. For example, you could connect the two phosphates not by a normal POP bond, yeah, that's just acid anhydride of phosphoric acid, but you make a PNP bond or a PCP bond by making an NH group between it, an imide group, or a methylene group between it. That one cannot be hydrolyzed at all, or only very, very slowly. These are stable analogs of GTP or ATP or what kind of other nucleotides you can get. By this, you can make also structures of these complexes which resemble the true structures, when the true nucleotide ligand is present, but which would not be stable enough that you could crystallize it. Here's a GAF, yes, guanine nucleotide exchange factor, also very large proteins. You see the SOS, for example, more than 1,000 amino acids. So you can imagine the full protein has a molecular mass of 120 kilodalton. And you can probably guess for what the rest is good for. Only one part is maybe required from getting the Rust domain between its tongues and squeeze around at its switches, switch 1 and switch 2, to release the guanine nucleotide to exchange it with the more ubiquitous GDP, right? But the other few hundred amino acids, they do interaction with all the other components of the signaling apparatus. So that's one here. Here's some comparison, P21 RAS with GDP, and what happens, with the SOS here, it squeezes in here in the site where the GDP is bound here, helix in, and to displace the GDP, and when it goes off, GDP can bind to it because it's much more higher concentration and restore the P21 RAS GDP complex. So I'm almost to the end for today, but I just would like to remind you that small G proteins and the structures thereof, also play some role in some other important signaling routes, the GPCR pathways. I will not tell you so much about it, but as you should probably have learned somewhere, at least at school, I don't know. When you think about our vision, yes, vision is done by rhodopsins, which is a so-called G protein coupled receptor that is activated by light. That is in working, this receptor works then as a guanine nucleotide exchange factor on the transducing complex by triggering the exchange of GDP against GTP. That's its primary purpose. That dissociates somehow, the trimer dissociates, and the GTP bound form of this G protein. Now it's not called anymore a small G protein because it's part here of a real large G protein with all the other subunits, but the one subunit with the GTP binds to the phosphodiesterase, and actually inhibits it, and by this, no, it's actually displacing the inhibitory component. Sorry for that. It's displacing the inhibitory component from the phosphodiesterase. The phosphodiesterase can then hydrolyze a lot of cyclic GMP to GMP, and when a lot of cyclic GMP is gone, then the cyclic GMP dependent calcium sodium channel cannot longer be open. The channel starts to close, yes, and that gives a change in the membrane potential, and that is transmitted by these neural cells, by the retinal cells to the brain. Yes, first to the retina, but later then to the brain. So that's the way how vision looks like. It works fantastically and fast. Yes. So it's a basic process that we do have here. Just to remind you a bit about it, G protein coupled receptors are nowadays an intensely studied topic in structural biology. Peter Kolb in the pharmacy department, for example, is using all the kind of structure information that is available to get some ideas about how ligands bind to it. Please remember that I think more than 40% of all pharmaceutical compounds have something to do with G protein coupled receptors at the end. Yes, they bind to it. So they are super important for pharmacy. Rhodopsin was the first one, but now we have structure of most other ones that also gave a nice Nobel Prize, a few years ago to the colleague who found the way to solve the structure solution problem for G protein coupled receptors. Here in rhodopsin, we have the ligand built in. So the ligand here is retinal, a chromophore that is absorbing the light. And it's just changing its structure from a 11-cis form, like this one shown here, to an all-trans form. And that one triggers this G protein coupled receptor to act on transducin. I will not go into details about this. Please read the textbook. I do not do it, just to remind you about the transducin. The transducin is a typical G protein, like GS alpha or GSI, the activating or the inhibitory G proteins that work in almost all of our cells. Transducin works, of course, only in our retinal cells because we don't need in other cells. So it also has the switch regions. Actually, the blue region shown here is mostly the small G protein-like structure that we have seen before. Yes, it's not different than the other ones. The red one is a kind of its own inhibitory component. It has to inhibit, the GTP hydrolysis. The beta subunit was this beta propeller subunit that is tightly associated with the gamma subunit, the membrane attached by some post-translational modification. So what happens when this one is exchanged from GDP to GDP? The complex just breaks here because the switch domain changed the conformation, and by this the interaction with the G-beta subunit is broken. And remember this inserted domain, the insertion domain, that is interacting with the switch 1 region, and the catalytic arginine is indeed part of the helical insertion domain. So what happens then after this, when the complex is released, yes, the alpha subunit with GDP is released from beta gamma. It can now do its work by binding to the inhibitory domain of the phosphodiesterase, but itself can now start to hydrolyze the GTP by its inbuilt insertion domain, which is not anymore inhibitory, but which starts now to put its arginine to the right side. So this is a time trigger. So only for several tens of milliseconds, this protein stays in the GTP-bound form. Then the time trigger protein, then it reverts back to the GDP-bound form. And this one we will switch, and I will also switch because I'm now at the end, and we don't need to have to talk about this, and I thank you very much for your attention, and hope I have some, when you have some questions, please ask them now. Hopefully somebody counted my I don't know this time, yes, but maybe it depends on time in the form, yes. Thank you very much. Bye-bye.